students, in its previous video we have studied about the turgid, turgidity, flaccid and flaccidity. In case of a cell to become turgid, the cell is required to be placed in hypotonic solution. Endosmosis takes place, the cell becomes firm. Now, in case of flaccid or flaccidity, when the cell is kept in hypertonic solution, exosmosis takes place. As a result, the cell becomes loose. It becomes flaccid. The flaccid term is used for the plant cell as well as for the animal cell. And we have also discussed earlier that a cell becomes flaccid due to transpiration, due to evaporation of water from the surface of the cell, it becomes loose. Due to less water supply, due to exosmosis, means there may be many conditions. But now we are going to study a special case for the plant cell only, that is called plasmolysis. Plasmolysis. Now we see that when a plant cell is kept in hypertonic solution especially, then across the cell membrane exosmosis take place. But the cell wall being rigid, non-living, freely permeable, remain in its place. As a result, the cell wall, the cell membrane will come on inside, leaving a space between cell wall and cell membrane, means the protoplasm shrinks. This phenomenon is especially known as plasmolysis. So, the flaccid term can be used for plant cell as well as animal cell if it will not be charged with water, but plasmolysis is especially known as known for the plant cell. Now, so what is it? The shrinkage of protoplasm from cell wall due to withdrawal of water or exosmosis, we can say withdrawal of water when a plant cell is kept in hypertonic solution. Hypertonic solution is called plasmolysis. So especially we see here, that is the cell, this term is mainly used for plant cell when it is kept in a hypertonic solution. The cell becomes flaccid or plasmolyzed. So in that case we can use both the terms for the cell, whether flaccid or plasmolyzed. Like you can see, suppose here there is a normal plant cell. If here there is normal plant cell, there is the cell wall and cell membrane. The cell membrane is in close contact with the cell wall. Now there is the nucleus, there is the large vacuole, and here is the cytoplasm, protoplasm. Now, if the cell is placed in normal solution, there will be no change in shape. Here there is cell wall and this is cell membrane. So we can see here, that is there is a cell membrane is in close contact with the cell wall. Now here, and now the cell is suppose placed in a strong sugar solution. This is initial condition. Now after some times we see that what happens to it. Again you can see, due to the exosmosis, as you know that the cell wall is rigid, cell wall is freely permeable, so it will leave the space Like you see, suppose here is the nucleus, and now there will be a small vacuole. We can mark here vacuole, a small vacuole. Now this one. So this is again cell wall. This is cell membrane, and here is the vacuole. Now we see, a student, that the exosmosis has taken place across the cell membrane, whereas through the cell wall bidirectional movement takes place. It is the freely permeable. The question is asked that what type of solution will be present in the space between cell wall and cell membrane. Here there is a shrinkage of protoplasm. So this is a protoplasmic contact. Here is the cell membrane and here is the cell wall. So even inside, that is in the space here also, there will be a strong sugar solution, or we can say that is there will be the presence of hypertonic solution in the space between cell wall and cell membrane. And this cell is said to be flaccid or plasmolyzed. So plasmolyzed word will be used only if the plant cell is kept in hypertonic solution and there is a shrinkage of protoplasm, leaving a space between cell wall and cell membrane. Clear, student? Now this type of diagram based question is asked. But suppose what happens if this cell, plasmolyzed cell, is placed in pure water, pond water or hypertonic solution? Then if the cell is living, in that case the protoplasm will absorb water, that is endosmosis will take place, and it will again become turgid. It will return to its normal shape. So if this cell is placed in, now if the plasmolyzed cell is placed in water or hypotonic solution, then in that case it will restore its normal condition or state. This reverse process of plasmolysis is especially known as deplasmolysis. So in the question it is asked that suppose there is the diagram given in the question for the plasmolyzed cell, and you will be asked to redraw the structure of the cell, the shape of the cell when it is placed in pond water. So we will have to draw this structure again. Clear, student? So this is plasmolysis, and what is now deplasmolysis? So we can define here again, that is deplasmolysis. Deplasmolysis, that is, we can say, the return of a plasmolyzed cell, not the general cell, it must be plasmolyzed initially, plasmolyzed cell to its normal shape when normal shape due to endosmosis when kept in pure solvent or hypotonic solution is called deplasmolysis. So this type of question is asked frequently, that is to redraw the shape of the cell and about the plasmolysis. We will study the different conditions or different applications of turgidity, plasmolysis and so on. Now the next part which we have to study here, that is about applications of turgidity. Now many processes are responsible are taking place due to the turgidity. Like suppose The first example we can take, that is the cell will increase in size. When the initial growth of the cell has to take place, like in meristematic tissue cell division takes place, its size has to be increased, then it is the turgidity that helps it to move. Now the first and the most important example, that is due to the turgidity root can penetrate into the hard soil, Now normally we see that if we have to penetrate a nail into the soil, it will not be pressed normally. But when a small seed germinate, the seedling grows in the soil, then due to turgidity, the root hair cells, the root gradually penetrate into the soil and spread over large area for absorption of water. Now, another example. Suppose a student we see here, that is the roots of some plants develop cracks in the crevices of old walls. Suppose if a small crack or a small gapping will be there in the old wall, and some seeds carried by the wind, by the air, will suppose germinate there. Gradually it grows, increases in size, and due to the turgidity, the root develops cracks in the old walls. So in normal condition, it cannot, old walls. So in normal condition, it will not be able to develop any kind of movement, cracks in it, but the root of some plants develop cracks in the crevices of old walls due to the turgidity. Now one more example. Now we see a student, that is in case of suppose we take example of drooping down of leaves of Mimosa pudica. Now there is this is the biological term, Mimosa pudica. The common terms for it, general terms for it is touch me not, lajwanti, chui mui. These are the different terms used for Mimosa pudica. Now this is the biological term. So first word is written with capital letter, second word is written with a small letter, and they are separately underlined. So drooping down of leaves of Mimosa pudica is due to the loss of turgidity. Now what happens in this case? When a leaf is touched, like suppose we draw a diagram of its branch. Now suppose here is the leaflet, and at this place here is the swelling, or suppose we draw here directly, here is the swelling. The swelling in the leaf at the base of the leaf petiole is known as pulvinus. So what happens here? When a leaf is touched, then the wave of chemical irritability passes down the leaf. As a result, the water from the cells of pulvinus move down through the xylem vessel, and the cells of it become flaccid, due to which it limps down, it droops down. you will see that when one leaf is touched, all the leaf gradually drop down. This is why it is termed touch me not or Lajwanti. But after some times, when the stimulus is over, again the water moves from root towards the leaves, the cells become turgid and it will become erect. It will return to normal shape because the cells become turgid. Clear, student? So these are some of the examples of this one. Now the next example is the opening and closing of stomata. Opening and closing of stomata. Now student, we know that on the surface of leaf, that is on the epidermis, there are the guard cells surrounding the stomata. Now how will it open and close? Now we see that in the cells of the epidermis, only guard cell contain chloroplast. So now suppose we draw the diagram of the stomatal apparatus. In normal condition, it has bean shaped guard cell surrounded by the other epidermal cells. Now this guard cell has inner thicker wall. This is inner wall, inner thicker wall, and this is outer thinner wall. This is a characteristic outer thinner wall. Now the guard cell suppose has, this is nucleus, and then there are chloroplasts. So on the surface of the leaf, that is in epidermis, only guard cell contain chloroplast, not the other epidermal cells. So during the daytime, photosynthesis takes place in the guard cells, producing glucose, which is soluble in water, due to which the cell sap of guard cell becomes hypertonic with respect to the surrounding cell. As a result, endoosmosis takes place. The guard cell becomes turgid and its inner thicker wall will bulge outward like this. Since there is inner thicker wall and outer thin wall, so when the guard cell becomes turgid, due to the turgor pressure, the inner thicker wall bulge outward. This is the opening of stomata. Now during the night, when the photosynthesis stops, the glucose from the guard cells is translocated to the other parts. Its cell sap becomes hypotonic, resulting into exosmosis, and hence the guard cell will return to its normal shape, closing the stomata. So during the daytime, due to turgidity, the stomata open, and during the night again it returns to normal shape. So the opening and closing of stomata is due to the turgidity. Clear, student? Now we have to further study here some applications of plasmolysis and some questions related to it. That is applications of plasmolysis. Applications of plasmolysis. First, jams, jellies and pickles are preserved by placing in concentrated sugar or salt solution. Now it is due to the plasmolysis, because you see that when jams, jellies and pickles, which contains the plant cell, is kept in concentrated sugar and salt solution, then due to the exosmosis, plasmolysis takes place and the germ cells are killed or they are not allowed to grow there. So, by the process of plasmolysis, they are preserved from spoilage. Clear, student? Now, we can take another example. Suppose in case of tonsil, we gargle with saline water or salt water. Gargle with salt water. What happens at that time again? Now, when tonsil develops, it is due to the accumulation of germs and it swells up. When we gargle with saline water, salt water, then salt water forms hypertonic solution around the tonsils. Due to exosmosis, germ cells are plasmolized and they are killed, as well as swelling is revived due to the exosmosis, and hence we feel comfortable. So this is the application of plasmolysis. Now, a student, a question is asked, reasoning is asked, that is, when excess of fertilizer is added to an unirrigated means without watering, unirrigated crop field, then crops die due to plasmolysis of root hairs. What happens at that time, you see? That is, when excess of fertilizer is added to the soil, unirrigated soil, then the fertilizers dissolve in soil water, moisture present in the soil, forming hypertonic solution around the root hair cells. As a result, exosmosis takes place and the root hair cells get plasmolized. Now, if this condition continues for a longer period, then the cells will gradually die and the crop is damaged. But if it is watered again, then the solution endosmosis will start and these fertilizer molecules will be used by the plant as nutrient, and then it will help to grow the plant. Clear, student? So we should not add excess of fertilizer or pesticides in an unirrigated crop field. Clear, student? Now, one more question. To clear lawn grasses, common salt is sprinkled. Now, what is the reason behind it? The same answer has to be written here. That is, when we add common salt into the soil, it dissolves in soil water, forming hypertonic solution around the root hair cells. Exosmosis takes place and the root hair cells get plasmolized, as ultimately it will die, and hence the lawn grasses will be completely cleared from the soil. It will not be able to regrow. Clear, student? So wherever we need to clear the lawn grasses completely, permanently, we add common salt to the soil. Clear, student? Now, so these are some of the applications. One more thing for the turgidity, we can write here application. Turgidity develops root pressure. Which helps in bleeding, guttation, ascent of sap, etc. So we'll study this topic with experiment in detail in the next part. Clear, student? Okay.